All right, good morning, everyone. So let's get started slowly with the general introduction. My name is Ahmad, so I'm representing the Center for Macromolecular Modeling and Visualization in Illinois, and we usually do these workshops to make you familiar and comfortable with the programs VMD and NAMD that we're gonna work and use this week. But we also go actually through the basics of MD simulations as a technique that is a very important technique in biophysical, modern biophysics these days, as well as so advanced applications of MD, namely in free energy calculations or very advanced modeling of complicated systems or enhanced sampling and things of that sort. So there is a good opportunity for you to really dive into these programs and run into troubles and troubleshoot them as you are here. So I really recommend the afternoon sessions, the hands-on sessions, what we call. So in the morning, we have lectures, and that's fine. People on Zoom can also listen. But in the afternoon, that's the real time when you apply the knowledge that you gained in the morning. So please, that's a very, very important part of the workshop. So as far as the program, so you have probably seen this, but again, day one, we're going to go through some of the basics of MD simulations and what is VMD, what you can do with NAMD, and so on and so forth. And then in day two, we're going to actually talk about enhanced sampling and free energy calculation. So we jump into the very advanced kind of applications of the technique, talking about collective variables. Some of our colleagues remotely are going to talk about that. Then on Wednesday, we're going to talk about force fields and actually some of the features of NAMD3, which is the new version of NAMD running on GPU and what it can do for you, what it cannot do for you, and so on and so forth. By the way, on Wednesday, we don't have it on the program, but we have a group dinner scheduled in the restaurant Piccolo, right next to the hotel, Auburn University Hotel, if you're staying there. So it's connected to that hotel. So we're gonna have around 6 o'clock. So we're gonna have a dinner, group dinner, and you're all invited, of course, to join you and your significant others. Lunches are also scheduled actually somewhere. Maybe you can, Rafael.
It's the dining, which is just a short walk from here.
Yeah, we're going to walk together to lunch, and so everybody again is invited. And we're going to have coffee breaks at 10:30 and in the afternoon, as well as breakfast, as you not noticed. So then on Thursday, we're going to again sort of talk about sort of some of the advanced, again, applications, network analysis, and Rafael is going to talk about actually very large simulation, very long simulation, and how to actually facilitate these. VMD2 is something else that is coming, and we would like to advertise this for the first time to you guys here. So that's the second version of VMD after almost 20 years, if you wish. So it's a very important kind of change and revision for VMD. And then you're going to also learn actually how to make really cool publication quality images with VMD. With a few tricks, you can actually take it from here to all the way to here to make it very high resolution and nice. And then the last day, we talk about the nanotechnological application of simulation and coarse grain simulation, as well as actually an interface that we have been working together with our colleagues in Georgia to make it easier for you to launch many, many simulations to get the results back, analyze them, and so on and so forth. We call it Cyber Shuttle. So that's something that you're going to learn on Friday. All right. So let me change my hat for a couple of minutes. So as the director of the center, so we would like to advertise about the importance of the technique. What you see on this graph, pretty simple. You see 3 bars. The first bar, the yellow bar, shows actually all the MD applications in sciences in general. So you can see actually there has been an exponential growth of the usage of the technique over many years. The blue bars show actually all the MD applications in life sciences, macromolecular, biomolecular simulations. And as you can see, they have been also increasing exponentially. And the red ones actually show the publications in life sciences that includes VMD and NAMD that you hopefully learn this week. So you can see that we are almost accounting for half of what is happening in life sciences. So these are very useful programs. And then they cover a lot of applications. You can see here, VMD especially actually is used in even quantum chemistry, of course, protein dynamics, and a lot of additional. This is based on the survey that we got from the users a few years ago. And in terms of the number of users, if you just look at the number of downloads of the program, we have more than 500,000 people downloading the program. But not all of them are regular users of the program. So we have been trying to narrow this down. For example, for the latest version of VMD, which was almost 200,000. And if we just look at people who have come multiple times and downloaded the program, so we have about 125,000 users just for VMD. And again, about 35,000, probably the same thing. But the point is actually it's a very popular program and it has a lot of features that you can use for your research. If you don't believe me, okay, these numbers don't mean anything. This is the number of publications. This is how much time it takes to get the next citation only to the 2 papers that we have, 2 or 3 papers that we have for these 2 programs. So right now, in less than an hour, in 53 minutes, we get 1 more citation. By the end of this class, we will have, I don't know, 20 more citations or more. 24 citations times 5, 120 more citations by the end of the program today. So people are using it. Many people actually, many more use and don't publish. Many publish, don't cite us. But if you just count people who publish and cite the programs, you can see that actually very popular programs, VMD and NAMD. So it's really good to learn them. Good. So now let's talk about MD and why we use MD. I'm just going to give you a general introduction here. So what are some of the areas, not all of them, of course, we don't have enough time. A week is nothing. So why we use MD? Obviously, computational structural biology, which has grown significantly by the advent of electron microscopy. So it relies on computing actually what is happening. And in parallel to what is happening experimentally, we have been also trying to improve the methods and the techniques to extend them to very, very complex systems, more complex systems than we used to simulate 2 decades ago. So you can see, so obviously you can recognize actually things that you cannot get experimentally, lipid-protein interaction as just one example. There is no method that currently give you this level of details of the system that you are interested in. You can look at actually conformational changes. We will talk about this a little bit more using some advanced aspects of MD simulation and see how things move. And that's something that no experiment can give you. They can give you the distance from one point to another point and a frozen structure, but they cannot give you the overall dance of the macromolecule. And the MD is something that you can give you that. Of course, there are approximations, there are computational tricks here, but if you couple it to experimental data together, you will have a very nice actually collection of 2 techniques. So computational structural biology is definitely one area that we look at. I usually use this slide when I did pharmacy 40 years ago. And when we studied pharmacy, there was no concept of where the drug binds to a receptor. We just had the pharmacokinetic, pharmacological properties, and they would give us the concept of pharmacophore. So there is this place, it must be like this. But nowadays, you can see actually this is a structure of serotonin transporter, a very important transporter in the brain. With an antidepressant drug bound to it exactly, you can see where exactly it's bound, what amino acid is it interacting with, what residue, what groups in the drug are interacting with it. And you have everything that you need to start looking at how this drug binding might affect the protein, the structure, dynamics, and function. So it's really a very rich area for the structural biology these days. And of course, I mean, if you want to design a better drug and so on and so forth, you can actually use MD simulations to gain more dynamical information about the system. So here is another example to look at the ligand binding that we did, I don't know, about 15 years ago, an ADP binding to a receptor. We had the structure of the receptor in an unbound form, and then we looked at the drug and see or ligand and how it binds to the system and how its binding is going to affect the residues in the binding pocket and so on and so forth. So we claimed actually this is one of the examples in which you see a spontaneous ligand binding captured by MD simulations. I mean, in parallel to our group, the group of Chris Schippo also actually did a very similar simulation exactly at the same time. We published them together actually. And then in addition to that, what you can do, what you wonder why this was possible in MD simulation. Usually it takes a long time for these things, excuse me, to happen. But we could capture them in hundreds of nanoseconds only. And the only reason was that because if we post-analyze the system, we realized that there is a significant positive patch engineered in this class of protein. Not only this one, the whole class of proteins in this family have this positive patch, and they all attract negatively charged metabolites, which is very common in mitochondria. So anyway, so if you extend your analysis of the system, you can really capture very interesting aspects of the system. If you compare this membrane protein in mitochondria with other membrane proteins in the cell, you can see that they have an extra 14 positive charges in them. So there has to be a reason for that. And that's the reason we can see you can attract ADP, acetic acid, anything negatively charged can be attracted by this. Here's another example. So a very important class of receptors, GPCRs. And we had the structure for the GPCR, but they didn't have it in the ligand-bound form. And the group of D. Shaw, so in 2011, a year after we published those papers, they looked at the spontaneous drug binding. In this case, there is no electrostatic attraction. But you can see MD can explore the phase space if you run it long enough. You can see how many microseconds the simulation is run. And after the ligand diffuses randomly in solution, at some point it binds the receptor and finds its binding pocket. And it's a saver. So you can say, hopefully that's going to be the binding site for the ligand. And very interesting, actually, a couple of years later, when we had the structure of this ligand with the receptor, this was right on the money, exactly the position that was found experimentally. So there is very, very strong, actually, possibility that if you run simulations long enough, you will be able to capture very important phenomena in the MD simulation by just running them. If you're patient enough and if you have resources to run these simulations long enough. So here is another case of a transporter that undergoes a large conformational change during its function. So there is, again, I repeat, there is no experimental data that can tell you what are the details of this dance. They can tell you that the distance between these 2 points, for example, changes, becomes smaller or larger due to ligand binding, but then you have to translate that to the whole protein conformational change. And that's something you can do when you combine MD simulations with the natural environment of membrane and lipids and water and everything with what we call collective variables that you can actually bias during the simulation, change the angle of, for example, helices with respect to each other. You have to design those collective variables nicely. And then you can see things happening. I'm not saying this is exactly the right thing, but at least you can explore them and see which one are energetically better or more compatible with the protein structure and so on and so forth, instead of sitting in one conformation and knowing nothing about the other conformation. So in the other case, we are monitoring the water contents of the 2 halves of the transporter, then they can see that they nicely empty and fill as the transporter opens and closes and undergoes the so-called alternate access mechanism of transport. So another area is nanobiotechnology, again, modeling of complex things. So here, for example, you can see that our colleagues wanted to actually look at the chip in which the antibodies are coated on the surface of the chip, and they're going to run a sample through it. And if there are viruses in the sample, you can detect them using this chip. So instead of using this cartoon model, so with MD simulation, you can actually put together this system, you can see the real antibody structures, which are not all kind of oriented in the same way, connected to the surface by PEG molecules. Some of the PEG molecules are collapsed on the surface because they are not connected to proteins. And you can actually, we haven't been able to simulate this system without water even actually is too large, let alone if you hydrate it completely and so on and so forth. So, but you can put them together very nicely using MD simulation. For example, using coarse-grained descriptions, you can allow them to avoid each other and especially arrange in a particular random way and so on and so forth. But my point is actually for modeling this complicated system, you can use the MD simulations for the different steps that are involved in MD in putting together a more realistic model for these systems. Here's another example again. So this is a gold nanoparticle that is coated again by PEG molecules. And then you add lipids to the surface. And this is the experimental view that we have, just a dot with a halo surrounding it. And this is the experimentally kind of published model of what this thing looks like. So this, I mean, this has no predictive power. Whatever you put there is there. That's it. So you don't gain any insight from these models. Whereas if you just use MD, for example, you can use the gold nanoparticle here, and then you can add the sulfur atoms that you need to connect to PEG molecules next, let them relax and run them, and then you can add the PEG molecules, as you can see, to the model, and then put it in water, simulate it, and relax it, and see actually how much water gets to the surface. You can see that actually water patches close to the surface of the nanoparticle, gold nanoparticle, for example. And then once you have a relaxed model for this pegylated nanoparticle, you can go on and add lipid molecules to complete the model to the surrounding and allow them to actually be absorbed by the particle, or you can even force them to, or encourage them, I should say, to connect to the nanoparticle faster during a limited time scale that you might have. So I would claim that this model on the right is much more predictive and more realistic compared to the cartoon that we have in the paper for this system. So here is another example. We are looking at ion conduction across a membrane through a protein. So initially, our colleagues actually couldn't even show that this protein forms an open channel. But once we put the green protein in the membrane, shown in this kind of ghost-like representation, you can see that these lipids can break the structure, can approach actually the middle of the membrane. So you see the head groups of the lipids are traveling all the way to the middle, are perturbed by the protein to complete the permeation of sodium ion. We can apply electric field, exactly like in experiment when you apply electric field to see how the ions actually are affected. And you can see how this sodium ion, this yellow guy, is going to be affected by the application of the electric field, and how the lipid head groups actually complete the pathway for the permeation of this ion from one side of the membrane to the other side of the membrane. So this is, again, I claim this is something that you never get from an experiment, because they cannot really get to this detailed view, and they cannot apply the electric field if you one day actually get to this detailed view. It's got to be a frozen system in which nothing happens. It's just a picture of the system currently. We can also look at actually much more complicated protein structure and even actually characterize them energetically, put a number to them. So like you can see the free energy landscape of this conformational change that we are showing for a complete protein in the membrane with substrate and ions and so on and so forth. So we will learn about these techniques more tomorrow from Chris and colleagues about collective variables, how to combine actually a group of atoms and put them all into one variable instead of just looking at one distance between two things. We can actually look at the distance between two helices or the angle between two helices. So that's something you can do with collective variables. So I strongly recommend you pay attention to this development because you will find it very useful for any kind of simulation, advanced simulation, free energy calculation, or enhanced sampling that you want to do. And finally, you will learn about QuickMD. That's we are trying actually to develop also interfaces that makes life easier for you guys. You don't have to go through everything and learn everything for as far as the setup of the simulation is concerned. You can use QuickMD. That's a very nice interface in VMD that allows you to take your protein, do whatever you want to it, put it in a membrane, put it in solution, check for the problems in the system and so on and so forth. And finally, you can actually even initiate your simulation and run it and do some basic analysis as is shown here. You can actually even interactively monitor the simulation and see how the RMSD is changing, how the system is evolving, and so on and so forth. So very useful graphical interface to the business. QuickMD, so you will learn about it more. As I said, actually, one very important application of MD simulation in which you're looking mostly at the dynamics of maybe single protein or a single system is how to put together very complicated things like a whole cell. This is the HIV virus, for example, the capsid only. So putting these things together actually is not trivial. So you might arrange them in a particular way, but they're not going to be stable. They might clash to each other. They might have large gaps between each other and so on and so forth. So in order to put them together nicely so they are ready for simulations one day, you have to use MD simulation for all the steps involved. So put one together to two, simulate it a little bit, relax it, and then add additional water or something like that. So in order to actually for NMR, X-ray, or electron microscopy to put these models together, they have been actually using MD techniques and classical simulations also. And finally, this is actually another application of MD that I find it very useful. So you can imagine you might have actually a structure of a protein, this might, in a particular state, in a state A, and then electron microscopy gives you a very low resolution of a state B, a different conformational state of your protein. And instead of just putting it there by hand and modeling it there, which is very tedious and very error prone, so you can start with this conformation A, and using MD simulation gently morph it into conformation B, so it actually matches the density that you get from your new experiments very nicely. Instead of just pushing it there, you just allow it with running MD simulation, relaxing other degrees of freedom of the system, put it inside the density and increase the cross correlation between the density and the proteins. We call it molecular dynamics flexible fitting. It's not just a fitting, it's flexibly fitting, allowing it nicely to go into the density. And many practitioners actually in the field are using MDFF as a tool for experimental biology actually these days. And here is actually the ultimate goal that we have is really to provide a graphical interface that does MDFF for you interactively. So maybe you have a very complicated density, you have to really do complicated things to fit the density. This interface allows you actually to use your pointer mouse or whatever and to grab things in the protein and to apply force in whatever direction you want to guide it into the density. Maybe it doesn't go there automatically and you have to help it. So you can see how this protein is going to eventually fit into the density that is shown in gray. And then on the side, you can see how the cross correlation changes due to your changes. You can actually guide your manipulation by monitoring what's happening to the system. So here you see the mouse, they're selecting actually what they want to apply the force. So then you can use the pointer. You can see the force as an arrow there. And again, cross correlation is shown here. You can see the cross correlation is increasing very nicely. You can rotate the system while you're applying the force. And you can see that that helix is nicely guided into the density while the rest of the system is doing its own natural motion and relaxing the response to what is happening to that manipulation. So that's interactive MDFF that we have in VMD if you have an application for it. And finally, this is another example of a very complicated system that we could put together. It took us several years, actually, to put it together. But the only way that I'm not going to go through the details, the only way we could do this was actually using MD for many, many steps. How to put the single proteins close to their surrounding lipids initially as a lipidated protein that requires MD simulation of individual proteins. How to distribute many, many copies of the protein in a spherical environment that required very coarse-grained type of simulations of these systems. And of course, eventually how to hydrate the system, you have to do simulation, fill the gaps and go simulations and so on. So these are just some of the steps that we had to put together to use to put together this complicated system with 1,400 proteins and millions of atoms. So there is no way you can do things manually when you're dealing with these things. So even actually when you put the lipids together, they're going to clash to each other. Cholesterol is going to form what we call the reinforcing type of interactions. And you have to have methods to check these and correct them automatically. Otherwise, you cannot do it by hand. I think this is my last slide for the introduction. Oh, sorry. But any questions? Please feel free actually to, yeah.
Are all the images shown in the presentation?
Guess what? Yes. Yeah.
Even the nanobiotechnology?
Yes.
Is that-
VMD is really powerful. I can't really emphasize how powerful VMD is and how many things you can do it. I mean, I don't feel I'm there yet myself after 25 years because I'm not really using it every day. But it has a lot of features, a lot of details that you can actually change and make your presentation or slide or paper very beautiful, actually. And you can engineer science into it. It's not just about pretty pictures and movies. You have to have a message, right? They say a picture is worth, I don't know how many lines of word, or no, a movie is worth a book. If you can engineer the science, the permeation of an ion from one side of the membrane to the other side with all the details shown, how the head groups are reaching the ion and so on and so forth. Yeah, everything VMD so far, yeah. Yes? Hi.
Can MD also be used to predict like timescales? For example, how long it'll take for a drug to bind and.
Can predict.
Yeah, can it also be translated to how long they'll?
If you run it long enough and if you sample the process enough, yes, because I mean, in molecular world, everything is stochastics, as we say. So they bump into each other randomly and they do things. Sometimes it happens in nanosecond. Sometimes it takes a microsecond. Sometimes it takes 10 microseconds. So you have to sample this enough and see what is the distribution of those time scales. In that case, you can, yes. But if your question is, can I maybe put a time stamp on it and say, okay, if I run it 10 nanoseconds, I'm going to see it? Unfortunately, no, you cannot do that. You have to try it. Any more? Yeah.
In your experience, how accurate are the energy barriers in the conformational changes of the protein?
Yeah, so this is also, again, I think as far as there are two sources of error for this business. One is the time scale. If you don't run it long enough, of course, you're gonna force the process too fast. You're gonna artificially get higher barriers against, because the system doesn't have time enough to relax and explore the phase space and find the best minimum energy path to do the transition. So that source of error, of course, depends on how fast or how slow you run the simulation, how long you run it. The other source is the force field that is not perfect. It's classical fixed charge usually. And from that perspective, I think we are in a good shape. We have really good force field these days. Despite all the things that are happening in the force field, they're pretty good. For example, for ligand-protein interactions, we are reaching within 1 kcal per mole from experiments, or even less in some papers. So, yeah. But for protein conformational changes, again, the force field is okay, but it really depends on how slowly and how carefully you do it, what collective variables you use, and so on and so forth. I'm going to actually talk about time scale, by the way, in my next presentation. That's the next thing that's very important for MD simulation and the ways people, some of the ways people have tried to overcome this problem, actually. Yeah. Any more questions from this side of the class? Yes. Thank you.
Any change over time, like 10 years or 5 years, of how, advanced the GPU
Oh, yeah.
power made these things a little bit more economical?
Absolutely, absolutely. I mean, GPU is probably, you mentioned it yourself. I mean, over the last 10 years ago or 20 years ago, when I started this, I published this, I submitted this paper to Science with 1 nanosecond simulation. And they said, this is not long enough, you have to do more. And we did 3 months of simulation, we made it 5 nanoseconds. Nowadays, you can run 5 nanoseconds in 1 afternoon with this, and even actually faster in a couple of hours. So definitely, GPU has been significant in change in the field. So NAMD3 that you will hear about is actually the main thing that happened in NAMD3 is to completely go to GPU, to be resident in GPU all the time and everything in GPU. We don't have all the features yet. We are working on it. That's the main thing we are doing. But the GPU has been the main actually event in the last 10 years, I would say. Yeah, of course, the algorithms are getting a little bit better and so on and so computers in general getting faster, but GPUs or everything else is here. GPU is here, really.
Interesting phenomena. It takes microseconds and stuff, and then it's
Even longer, yeah.
To do this, but it's expensive.
Absolutely, yeah.
If it's changing,
I guess anybody who solves this time scale issue with MD simulation would get a Nobel Prize probably. That's okay, so let's see. So let's talk about time scale a little bit. So that's one major limitation in this business. So do we have it here? Yeah. All right. Do a few things. Like this again. Okay. So this is a 5 nanosecond simulation I submitted to Science, by the way. And this movie made it to the Nobel Prize website, by the way. It was 20 years ago. And that's the closest, I joke, that's the closest I get to the Nobel Prize, I think. But this movie actually shows you how a single file of water is crossing the water channel across the membrane. So we publish it together with that science paper. So, but it sort of captures a nice process because it does happen on a nanosecond time scale. So we can nicely capture it. But I would say there are 2 major limitations to the technique. One is, again, we discussed it already. One is about the time scale, which translates into limited sampling. That depends on how fast your computers are, how many GPUs you can run on, how patient you are, how much money you have, and so on and so forth. So time scale is a major issue. And the reason for that is that for capturing these processes, you have to run the simulation on a femtosecond time scale usually. Every step of the simulation is femtosecond, 1, 2, 4 femtosecond. And that means for a microsecond of simulation, you have to have billions of steps of calculating forces and interaction between everything and so on. That makes it very, very expensive, and it cannot be parallelized that easily. You have to take a step 1 before you can just take a step 2. And so on. It's a serial problem. So that's why that remains the main sort of issue with classical simulation or any type of MD simulation that we do nowadays. The second problem is actually the force field approximations that you have. For example, all these charges representing these water atoms are fixed. Oxygen has a fixed charge on it. Hydrogen has a fixed charge on it, which we know is not correct because when they come close to negative or positive things, they're going to be affected, they're going to be polarized, and the charges are going to change, and that's going to affect the dynamics of the whole system. So we don't have that description, for example, here. So this is one way, and of course, the forces that we developed are coming from a particular configuration or conformation of the system in which we might not have actually representation for other conformation of the system. That's another source of error, for example. So even though we have this problem, but still, as I said, the forces are pretty good, relatively speaking. The main issue, I would say, is the time scale, and the second issue would be force field approximation. But you should have these things in mind when you're interpreting the results of any simulations and see, for example, if there's a conjugated system that can be polarized heavily due to the protein environment, then you have to actually keep that in mind as a source of error in your MD simulation, or maybe modify the force field a little bit for that special case to be able to get a more accurate result. Despite all these disadvantages, I would claim that there is no other technique in science that can give you unparalleled, spatial and temporal resolutions simultaneously at this level. 1 femtosecond resolution, and you can keep track actually of the position of every atom every 1 femtosecond or every 2 femtosecond during your simulation. There is no experimental technique that can do that, right? It even defies the uncertainty principle, actually, right? But here you can even go down, go to attosecond if you want, right? No problem. So that's a very powerful aspect of it. And again, as I repeated, but of course, everything is virtual. Everything is in your head or in your force field world. It's not God's world. So you have to somehow anchor it into reality by combining these results to experiments, going back and forth. Then you can have actually really nice results and discoveries. Okay. So, overcoming the time scale limitation, as I said, that's a major limitation in the business. So we want to system, for example, a transporter system goes from outward facing to inward facing on this sort of simplified energy landscape. But you can see it's a very rugged landscape, and there is a minimum energy path you can recognize, but still the system which has to randomly explore the phase space completely almost with other degrees of freedom to be able to travel over many, many microseconds or milliseconds or even longer to the other side. So there is almost no way that your protein does that in regular MD simulations. So you have to do something for it, at least these days, to be able to describe this process semi-accurately and then see how much more you can relax the system and so on and so forth. So you have to somehow enhance the sampling for the system. And I'm going to talk about a few of the things that have been developed maybe over many, many years and how people approach this problem. The first thing that people did was the so-called coarse graining. So instead of having the position of every atom in the system, I can actually put maybe some of them together and have a bigger bead. I'm not going to call it atom or particle, you can call it, that captures actually the behavior of that whole thing as one group. So you're going to lose some resolution, of course. You're not going to have perfect hydrogen bonds anymore. But then these big things can actually move more softly, and you can take longer time steps and so on and so on. For example, for the case of lipids, this is a simulation that we did a long time ago, and it shows actually 5 nanosecond simulation of a pure POPE. You can see all the lipids, they move up and down, but they're not going to go anywhere. You don't see any mixing of the lipids or any exchange of the position of the lipids. They're just moving up and down, changing the thickness a little bit. That relaxes relatively fast, but there is no exchange. That's a kind of becomes very important because these days we are trying to simulate heterogeneous lipid bilayers around proteins. So however you place them initially, they're not going to go anywhere. They're just going to go up and down a little bit. So you don't see mixing around the protein, how they might accumulate around the protein, and so on and so forth. So just to coarse grain this as an example, so we can now, instead of having the full lipid with all 150 particles, I can coarse grain, for example, the whole positive charge in the head group as one bead, negative charge in the head group as one bead, glycerol moiety as one bead, and every tail as 3 beads. So beads are usually spherical. I'm just putting them like this, like oval shape, to match the shape of the lipid that you have up there. But by just doing that, you can see you can decrease one order of magnitude or more the number of particles that you have to calculate forces for. And more importantly, because the motion of these things are not going to be as fast wiggling of those small things, so now you're not limited by that 1 femtosecond. I forgot to mention that you take that 1 femtosecond time step because otherwise your simulation becomes unstable. If you take longer time steps, your atoms move too much, run into each other, or get too far from each other, and then you will deal with even larger forces in the next step, and your simulation is going to crash. So depending on the vibrations of the system, the natural vibration of the system, you have to tune the time step that you take in the simulation. So by going from very small particles, fast particles like those, to these sluggish particles, you can increase the time step. Instead of 1 femtosecond, you can take 20 femtoseconds, 40 femtosecond time step. Then you can reach much longer processes or time scales describing processes that you're interested in. So this is a simulation formation of a lipid bilayer that actually took from the article from YouTube channel of Axel Kohlmeier. So you can see it's initially starting with a coarse-grained representation of lipids randomly placed in solution. And they eventually form a bilayer during the simulation by finding each other slowly, and so on and so forth. So this coarse graining has been, just for your information, very powerful and very useful for lipid type of structures because you essentially deal with two types of interactions, polar interactions in the head group, hydrophobic interactions in the center, and you can really nicely distinguish between these two with this coarse graining model. For proteins, less successful because you are dealing with 20 different amino acids, hydrogen bonds, a lot of additional interactions that have to be taken into account in order to describe the protein structure and dynamics. So I would say relatively you will see more coarse graining for things, simple things like lipids, formation of liposomes, liposome merging, fusion, and so on and so forth. So yeah, so this is one way of actually approaching the timescale problem. So now the other way is actually maybe this is something that we did few years ago to reduce or simplify the representation that you're working with, for example. So in this case, we are interested in how proteins bind to the membrane, to the surface of the membrane. So we argue that maybe in the center of the membrane, the core of the membrane, you don't need to have these long tails that make the lipid sluggish. We can cut them and fill that area with the solvent. So we called it terrible acronym, HMMM. So I can't change it anymore, unfortunately, it's too late. But that allows us to capture the binding much faster. The initial binding, we can capture it very fast. So the problem is more described here. So if you do this atomistically, you see some initial binding, but there are some problems here. Usually you have to apply a bias and push the protein into the membrane. You don't know how far you have to push the protein into the membrane. Does it be 5 angstrom or 15 angstrom? You don't know the depth of insertion in this case. Almost frequently it fails. It doesn't work completely because you don't have enough room for the protein to be synced in. And then you cannot repeat it because it takes a very long time. So we published that single membrane bond structure in a paper, but we were never happy about the results. And of course, the lipids don't reorganize. That's a problem with membrane. So we, as I described, actually developed this HMMM system. We replaced the tails with an organic solvent. And then what's going to happen is that actually you can see that when you simulate them, the surface of the membrane, if you look at the distribution of different atoms, the surface of the membrane looks very similar to full membranes. So water and everything is, their distribution kind of match very nicely. And then if you look at the diffusion constant for the lipids, lateral diffusion constant of the lipids, how fast they move laterally, you can see that actually here we are with the full membrane. We have these two for different area of the lipids. And then this is logarithmic, by the way. And then if you go to HMMM, you're going to increase 1 order of magnitude the diffusion constant of lipids. And you can even do a little bit more trick and maybe make it lower density, make sort of expand the area and increase the area per lipid and gain another order of magnitude increase because lipids are not far away from each other and can move even more. And what does it mean? So that it means that here is a comparison of 10 nanosecond simulation of a traditional conventional membrane. And as we discussed, lipids are not going anywhere, they're just beginning where they are. Comparing to a 1 nanosecond HMMM membrane in which you can clearly see lipid displacement and mixing during the simulation. So we can really expect to have a nice heterogeneous membrane. Maybe if I have a protein here sitting in a HMMM, then lipids can actually explore the protein. Maybe you can accumulate PIP2 or cholesterol or other lipids around your proteins. So, but again, so this system allows you to capture the simulation, the binding process very fast. This happens in actually tens of nanoseconds only instead of running it for hundreds of nanoseconds because lipids are more mobile and they're more accommodating. And because it's fast, then you can have n in your paper, n equal 10. And you can see that actually nice, we have also nicely analyzed them and monitored how the center of mass of the protein gets close to the membrane. And you can see that, oh, the center of mass of the membrane binding domain. And you can see that many of them explore the solution, but they all nicely converge to the same level of insertion into the membrane. So now we are feeling very confident about this level of insertion into the membrane. So of course, you can always take this. Once you have reached your results, you can convert it back to a full system, to a full membrane system, for example. You can do the same thing for the coarse grain simulations also. Run it coarse grain, explore the phase space a little bit more, and then sample from your initial coarse grain simulation, convert them back to atomistic simulation if there are atomistic problems, and see what happens there. Yes?
Can you explain what you mean by replacing the lipid tails with an organic solvent? Is it still an atomistic model?
Yeah, it's still atomistic, yes. We take
some organic
it's just
solvent.
organic liquid. Instead, we have a complete fluid instead of having those tails that are entangled to each other and prevent each other from move. So it's still atomistic. This is not a coarse grain system, but it's a reduced representation with an increased diffusion constant for the lipids. The head groups are completely, because if I go back to, the head group is completely the same. Just the tail part, the yellow part is, because the protein doesn't even go there. The protein just binds to the surface. This membrane maybe all the way to here. So there is no connection to that part of the system. So it's perfect actually for peripheral membrane proteins because they don't interact with the core of the membrane. But nowadays we are extending its application to transmembrane proteins as well. Yeah.
You show that when the protein binding occurs to the lipid, that there are some ionic displacements from the head groups, like the solvated ions get displaced into solution. How well does MD capture these kinds of entropic contributions?
Very well, I would say. Yeah, that's the first thing MD does for you to randomly move these things about. And if you, again, it all goes back to the time scale. If you run it long enough, MD is able to nicely describe. For certain ions, for example, magnesium has an inherently very slow kind of dynamics. So it takes a microsecond for magnesium just to change the hydration shell in water, nothing else, no membrane or nothing. So now if you put it next to, I don't know, a protein and it sticks to a carboxy group in the protein or DNA, nucleic acids, then you're gonna have to wait a long time for it to go back and come back binding to something else. So those things, divalent ions are difficult, but for sodium, monovalent things, chloride, potassium, and things like that, they're pretty good actually, yeah. Yes?
Is there a function of wisdom to decide the size of the preserved head group?
Oh, here in this, we tried 5, 6, 7, 8. And then this was actually what I don't remember, we are selecting 5 of them actually, we are keeping only 5 carbons in this case. So it was the best kind of qualitative comparison between the atomic density and the full membrane system. At the same time, approximate enough to give us increased diffusion constant.
This method should work for heterogeneous membranes.
Yeah, absolutely. Any phospholipid membrane we can do. We just published actually last week a paper in which you can add cholesterol also, because cholesterol you cannot cut, it's a bunch of rings. So, but for phospholipids it's easy. Sphingomyelin we are doing, POPE, POPS, PIP2, those are easy actually, you just cut them. That's it. But for cholesterol, we had to apply some tricks to keep the orientation of the cholesterol, because otherwise it's going to fly into the liquid phase. That's a very nice environment. And you don't have that cholesterol arrangement in the lipids. Yeah. The more questions, the better, guys. Sometimes I even say we don't have lunch until you ask 10 questions. Yes, Sudhakar.
So, I mean, of course, you're replacing the chain, lipid chain with individual lipid-like moieties.
Yeah.
And, okay, so whatever, of course, it increases the mobility and so on
Right.
and so forth, but what are we missing? And so in nature, they're already connected and so on, right? Also, are you able to actually do this displacement of these head groups from one position to the other? Beyond what you observe in a chain, lipid accumulation? Or is it something else that is just speeding up the insertion?
No, actually, I mean, because the lipids move faster, they can make room for an incoming protein much better and faster. That's it. And as I said, we are using 5% increased area for them initially as well. Every time you look at membrane insertion. But I think the fact that the tails are not entangled down deep in the core of the membrane, because if they want to displace and change position, for example, the tails have to follow, right? They are entangled to each other. They can't do it fast enough, right?
In nature, they are doing.
Yeah, the nature has time. You have to, you put it there and come back next hour. Good, no problem. I guess if we reach the time scale that we want, we don't have to do all these crappy tricks, actually. So these are all coarse-grained even. It's terrible if you think about it. But we have to do it, otherwise you don't see anything. That's the problem. Yeah. Yeah, nature has had how many billion years to get here? So yeah, anyway, so the method we have used is for many peripheral problems. This is one example of how a fusion peptide of a virus, I think this is flu, influenza hemagglutinin fusion peptide binds to the membrane to initiate the fusion process. And again, I don't want to, we have had a lot of applications for these things, but one point I want to make here is again, another way of maybe increasing your sampling of the space is to maybe randomize the initial configuration a little bit. So here we are placing the peptide initially in 7 different initial orientations, upside down, this way, that way, and so on and so forth. Instead of waiting for them to explore those things, we can maybe help it by initially actually randomizing the configurations. And then very nicely, actually, we captured the binding to the membrane using this model in less than 100 nanoseconds in each case. And if you compare the result of all of these simulations, initially started with different orientations, you can see they all converge. This is a top view. In one case, so unfolded, but in other cases, they all converge to the same configuration, even though initially were different. And that's the side view, how they sat on top of the membrane at the end of the simulation. So we have very nice convergence of the results in this case. We can even actually look at the tilt angle of the helix in the membrane. So you can see we initially placed in 40 degrees, 20 degrees, 0 degrees, 10 plus, 20 plus. And they all nicely converge. Actually, you can see they change their tilt angle and they reach actually the same orientation at the end of the simulation. So yeah, so we use this method. This is another example of how a full cytochrome P450 sits on the surface of a membrane. And upon this, they can recruit its substrates from the membrane into its binding pocket, which is right here. So we should, actually, this is the best way for the protein to recruit its substrate, not from the solution, from the membrane. And by the way, this protein is also anchored into the membrane with the helix. So there is a linker that connects to a helix, and that helix is sitting permanently in the membrane. So the protein can sort of go away a little bit from the membrane and come back. So we did this, and again, you can see we started with different orientations, different tilt angle of the heme with respect to the membrane, that's the heme part. And then you can see that actually we are converging to a value that is within 10 degrees of experimentally measured. In this case, they can calculate actually what is the angle between heme, because it's photoactive, and the surface of the membrane. So the value they calculate is within 10 degrees of the fluctuations that we get at the end, even though we start from different initial configurations of the system. So I'm gonna skip this. Okay, so the next thing that we can do to make sure we have time, and please interrupt me if you have any questions of any part, so don't let me talk too much. So the next thing that we can do is called the steered molecular dynamics. People have different names for it. Essentially, the point is, in addition to the natural forces that you calculate from your force field, you go in and add additional forces in a particular direction to your system, to a particular group of atoms in the system. So a steered MD simulation, so as I usually use myself as an example, let's say you're tired, you want to see me leave the room, but I'm just here diffusing randomly like a blind or drunken guy, just not leaving, and you're sitting here weeks and months, I just maybe approach it, come back. So you can't wait for this long enough, so you can add a very tiny force on me to one atom or a group of atoms, or hold my arm to encourage my still random, I can come here, but my movement toward the door is going to be increased by this force that you add to the system. So you need to know the direction that you want to apply the force and to what group of atoms you have to apply the force. Otherwise, for example, if you apply too much force to my shirt and I'm heavy, you might tear apart my shirt without actually completing the process. So these are things that are a little bit of an art. You have to learn how to do these things. But by doing this, what we call the steered molecular dynamics simulation, I still do my random fluctuations, but they are biased for the particular direction, this door or that door. And then another thing is the direction. So how you're going to apply this force when you come to a more complicated group of atoms. So maybe there is a cable in my way here. Maybe you want me to leave that door, and you cannot apply the force like this. I will go over this table. You will get unnatural kind of pathway for me. I have to go over several tables. So I have to maybe come back here, go there, and then finally reach the door from the end of the room. So you have to really experiment with the system and decide about the correct collective variables, how to take me to the other. Maybe I should go to the ceiling and come back down to the other end. So it really could be very complicated in multi-million dimensions that you have in a molecular system. So that's a very, this kind of sort of in general bias type of simulation, and enhancing the sampling of the space is really a very big field, and you will learn about this more tomorrow. So here is an example. For example, we have two channels. Both of them are water channels. One of them is also glycerol channel. And we were wondering why that one is only good for water, why this one is so good for glycerol and water. So what we did was, we can tell now that we have the results that the size of the system is larger. But essentially, we wanted to look at the process. We decided to look at water permeation happens spontaneously, as I showed you in the movie. in the Nobel Prize movie. But the reason is that you have a very high concentration of water. What is the concentration of water? 55 molar, not 100 millimolar for an ion or something. 55 molar, you have tons of water in the system, so you have a chance for some of them to make it. Plus water is very small and diffusion constant is high, and it can actually really does the process spontaneously in a few nanoseconds. For glycerol, it might be actually a millimolar concentration, 1 or 2 millimolar in the system, process big. So it's not gonna do it spontaneously in my 10 nanosecond simulation. So what I'm gonna do is gonna do the steer trick. I'm gonna connect to the center of mass of this guy or to one of the oxygens or to the middle oxygen. You can decide what kind of process you want me to rotate and go this way or go this way. You can decide about the process you want to simulate. And then apply an additional force to it. And you can describe, my plan is to describe glycerol motion through the channel that goes through, and also take it through this channel and see where the barriers might be. Why this is not a glycerol channel? What are the energy barriers in the process? So I can do that and then monitor the position of glycerol. There are different schemes of applying this force. You have, if you know in advance what kind of force you need to complete the process in your 100 nanosecond simulation, then you apply a constant force. But usually we don't know. We don't know how much force, 10 piconewton, 100 piconewton or more. So therefore we use something we call constant velocity, SMD, but it's a variable force scheme in which you increase the force by a trick until you overcome the first barrier and the second barrier and so on and so forth. So how do we do this? We are gonna connect to me, instead of pulling my shaft directly, we're gonna connect the spring to me and then pull the other end of the spring. So as you can see, you're gonna increase the length of the spring and increase the force until I start cooperating, and then you can bring it down. So in this manner, you're gonna apply the necessary force, not going beyond the necessary force too much. So we call it constant velocity because you pull the other end of the spring with the constant velocity, and you can calculate from here to there is 5 meters. I can run 10 nanosecond, 5 divided by 10. So I have to pull this guy 1 meter per nanosecond, or 2 meters, or half a meter per nanosecond to complete the process in 10 nanoseconds that I can run on my computer in the lab. So you can adjust the pulling in that manner. I guess you're going to talk about SMD a little bit more, Rafael. And yeah, so Rafael is an expert in SMD, multiple papers. So you can actually, again, look at what happens as I said, this is a trick to let a process happen. Because if you just wait for it, it's not going to happen. You can decide, so okay, this is not good for simulation. I cannot make any conclusion about this particular process. Why? Or you can say, okay, I'm going to apply SMD. It might be hysterical. I might be going over some tables on the way. But at least it happens, and I can explore the system. Maybe you can take me from this top of the table, simulate me here, and see how I relax locally to a low energy state. Kind of. So you can explore the path a little bit.
I have a question. I'm kind of confused. So if we want this process to be happening and we start, applying forces to the object to, for example, to you to get out of the room. And if we do it, from the beginning all the way to the end. So where is the surprise? So we applied all the forces. Is there any point that we should stop the force so that, like, okay, we have the right conformation for example, for the system that we study, and at that, and when it reaches that conformation, there is no force needed to actually finish the process. Is that such a thing, or?
You can do those kind of things. You can actually take me halfway and let go of me and see if I snap back or continue the process and so on. But the whole point is actually to see that I walk this path and what happens to me. Maybe here I just interact more with the screen, hydrogen bonding here, maybe I shake hands with this guy first before we, so all the process is happening along it, because other degrees of freedom, I can do everything as I'm doing this movement, right? So you can explore those aspects, those are the surprises that you're asking for. But yeah, so you can do the other thing, you can sample the system and then take the samples and then run additional simulations on those samples and see how they locally behave and so on and so forth. Yeah. Yes.
Explain what the sharp drops mean.
Oh, I see. So yeah, so this is the Z position as the glycerol moves through the channel, and that's the time. So during the simulation, as you're pulling it this way. As you can see, so let me show it with your pointer. So as you can see, initially sort of it comes into the channel nicely, but then it hits a barrier. It stays there, and you have to increase the spring length to accumulate enough force to get it over the first barrier, and then sticks to the next binding site, kind of, and so on and so forth. So you have these transitions between one side to another side. It could be hydrogen bonding to the maybe other groups in the protein. It could be a narrow point of the protein that you have to squeeze to go through and so on. Could be steric, or it could be chemical interactions, kind of thing. Yeah, that's another thing. You can explore these binding sites or sort of minimum energy positions by just taking them from this analysis. Here we are showing, for example, what kind of hydrogen bonds are happening in that spot. So you can take that guy and just simulate it by itself and see if it's stable there or yes.
So is there any notion of this differential, partial pressure or something like that, of the water, in this case, we're going to do the glycerol, the single molecule.
I'm very glad you asked that.
Speed
Yeah,
gradient
yeah.
that mimics, or we can mimic for the force, or is there
I
a
see.
relation between those two?
So usually we try to, I'm very glad you asked this, because if you just do SMD without doing anything to the system, if you try to pull me alone, I'm going to take the whole class with me, actually. The whole membrane and water and everything moves. So you're not going to have an effective translation. You have to fix something to the system or apply a counter force to the system to keep it in place. The center of mass of the class should be kept fixed, and then I will be pulled in that direction. Does it make sense? So the total force to the whole system is going to be balanced, and there is no additional force to create a pressure or anything like that. Did I answer your question? I feel not.
I was thinking, this force that it has a correlation in terms of something like a gradient.
I mean, at this scale, not really. I mean, you're applying a force to maybe 6 atoms in a 100,000 atom system. But we have used actually the district to create a water gradient actually by applying this kind of force to many water molecules in a particular slab above the membrane to create a gradient. And then you can encourage the water movement from one side of the membrane to the other. So otherwise, it's going to be randomly going up and down, right? Especially in a periodic system. Or ions, yes, yes.
I mean, you can also apply some kind of an
Yeah.
field gradient.
Yeah, the electric field. We can apply electric field. Electric field is exactly SMD for charged species. Yeah. And you can define only on ions. I don't want it, this protein will be affected and so on. Yes. Yeah. Okay, let me skip this. This is, but again, we can energetically describe this and show that for the channel that conducts glycerol, the energy barriers are very low with a few kilocalorie per mole, whereas for the guy that doesn't conduct ions, you can see there are huge barriers in the middle 2 or 3 positions. And of course, if you look at them, you can see that it's very narrow in those regions, and that's why it causes this. But again, I think the whole point is that you can quantitatively describe your system very nicely and put some numbers to the calculations that you get. Okay, so Case 3, now you can take SMD or a steered or biased simulation and apply this instead of to one atom or a small group of atoms, you can apply them to actually complicated groups of atoms and describe very complicated type of motions in the system. For example, here in this blue-red case here, on the right side, so I'm going to change the orientation of these two halves of the protein like this, change the angle between them using what we call collective variables. Cobalt is a very, very important thing for you guys to learn tomorrow. And by this cobalt, you can actually rotate things, change their angle, apply quaternion changes in the system, or simple translation and orientation change, and so on and so forth. So the same idea, you apply force to the system, or you bias the value of a particular collective variable in the system by using a kind of SMD-like approach. But again, instead of one atom, you're going to use a group of atoms, and then you can, for example, change the helicity of this helix. You can go from a fully helical form to a completely unfolded form, or half helical, and so on and so forth. So these collective variables are very powerful. You can mix and match them very nicely, and you can do really any trick that you want to the system by designing a nice set of collective variables. You can have multiple collective variables, one of them moving very, very fast, one of them moving very, very slow in another direction, and so on. And by doing these combinations, again, you can engineer any type of translation, manipulation to the system that you want to explore and energetically describe later. So we will learn a little bit more about this tomorrow. The whole idea of this transporters is that actually, if you take a linear path from beginning to the end, so this is what we call targeted MD simulations. You go from one side, one valley to another one, probably over a barrier, over a very high barrier. And even if you relax it a little bit locally, you might be way too far from the actual natural transition pathway in your simulations. So this is a map of Telluride. If you have been there, you have to take a car if you want to go from Telluride to Monterey Village. On the other side, you have to take the path like this. That's the only road. But you can take the I forgot the name that takes you up the mountain. Yeah, you can take it and then go up the mountain. So that's what happens in TMD. But there is no chance that you get an idea about the natural pathway if you take that. So that's why our approach actually for this process is to use many collective variables, because we don't know what is the collective variable that's going to take me from here to there. Again, I repeat, so I can go that way, I can go this way, I can go to the middle and come back. So I have to really explore the space a little bit more. So we are using many, many collective variables. We look at the protein structure and say, okay, this seems that these helices went from this orientation to that orientation. So let's try to change individual helices, or as a full domain, we change the angle, or we change the center of mass at the same time, center of mass distance. So we're going to explore the phase space using many, many different pathways that we engineer artificially. And if we do that, then we somehow quantify them, use non-equilibrium work to see how difficult it is for the protein to undergo these transitions. It has a lot of dissipative work in it, but still you can get some ideas, and you might get close to the natural pathway as parts of this exploration. And then we take those and try to relax those into natural pathways. It becomes a very exploratory kind of approach, but then it gives you much better answers compared to a simple SMD-based or MSD-based type of targeted molecular dynamic simulations. So in a sort of, again, so we can see that here we're going to bias. A collective variable could be anything that I did sort of mention. So you will hear more about this. And compared to the initial state and the final state, we know the values of these things. So we're gonna guide the system using this collective variable to go from initial system to the final system. It was the initial system value. This is the difference between the initial and final. That is multiplied by the time that we need to simulate to describe the whole process. And these collective variables can be anything. Could be radius of gyration of the system. Very nice. This is, you can see, how am I going to go from this configuration to this configuration, but just R of G. It's a very powerful way of actually manipulating the system. Let me skip this and just show you one of the first applications of this type of approach, how we describe opening and closing or OF-IF transition for this particular protein, but for this ABC transporter. But then next, we took actually the next step for this antiporter. You can see that here on this leg, we are actually describing how it goes from outward facing apo to outward facing bound. The substrate is bound here, so we have to describe it using this different set of collective variables. How we describe the transition between the allowed path and disallowed path using actually the helical orientations, and the binding event on top, again, is described similar to this. And by doing that, actually, we could actually show why this path without the substrate is not allowed, and why this path with the substrate is allowed. And that's actually the physiological mechanism of this antiporter, because if it wants to antiport, it should not be, you should be able to only complete the transition when the substrate is bound. You bind something, bring it to the other side, you bind something else, take it to the other. This coupling between these two processes is only preserved because you cannot undergo this transition. Otherwise, you bring it down, release it, come back this way, and bring it back down. And that's not coupling between transition of one species to one side of the membrane and the other species in the opposite direction. So it's very important, but now what can we do? So this is now what I mentioned about calculating the free energies of the system. Now we can use these collective variables along a particular pathway and see what happens to your energy along the pathway. You can see the red one represents the case for the apoprotein when the substrate is not bound. And you can see there is a very large barrier there preventing the system from undergoing those conformational changes. Whereas when the substrate bind, of course, you come to a different state bound compared to empty form. And the barrier separating these two is going to be smaller compared to the apo case. So, but again, the details don't matter. The point is really you can take all these highest type of simulations or pathways that you discover for your process and then take them to the free energy level to put a very quantitative number to them and compare it to experiment directly if they can measure this. Yes?
What exactly is the imaging? Imaging.
Image. Oh, okay. So we had to break the paths to images. You know, every image represents, for example, you can separate them by, I don't know, depending on the collective value, by 1 angstrom at a time. So you call it that one image, and then you take that image and locally relax it as much as you can. And then until you converge. You can see the standard errors are actually pretty small in this case. Yeah, that's what it is. 100, 150, yeah. Okay, so that's my presentation. Now questions. So this is general introduction to why we do MD, for what purposes, why do I need MD simulation, what can I do with MD simulations, what are the major limitations, what people have done to deal with these kind of limitations, some of them. I mean, there are many, many more things that people have developed over years, but we don't have time to talk about them. And maybe some of them you can get from other talks, and some of them you can get from your exercises. Yeah, question. Then we can have a coffee break. Yes.
So, where does accelerated MD falls in this situation where you try to reduce local minima to reach a faster scale?
Where is the accelerated MD force?
Yeah, no, I mean fall. Where does it fall in this?
Oh, in this scheme. So in this scheme, the whole transition. So going this guy, taking this guy from here to the other side. You see, even in the allowed form that happens in nature, it's going to take milliseconds, and I cannot simulate that. I have to force the transition. But how are you going to transition? Force the transition. I repeat my own example. I want to go from this state to that door state, back home, back in the class. So I can go directly, I can go up and come back down, I can go through the floor, turn around, I can go here and then left, I can go there and right. And you can imagine a multi-dimensional case like this protein, it can be very complicated. So you have to decide what is the pathway that you want to simulate. And then based on that pathway, design the collective variables that take you along that pathway. And then this become the images that I mentioned to him. So image 1, image 2, image 3, image 4, and so on and so forth. Then you can take these points, do a lot of relaxation that in which case, that we did in this case. And after those relaxations, you can get the free energy values for them. The force is applied to induce the process that you are trying to describe with empty simulation. Otherwise, if I just simulate it here, I'm going to just capture it for the whole 100 microseconds or so. Yeah. 1, 2, 9 more to go before the coffee break.
Talk about like any mutations or something. So what happened is not necessarily the mutation leads to structural changes or any conformational changes, but we do see changes in the function.
Yeah.
So how like in we can use the simulations and MD simulation to understand that molecular mechanism, which we can't see in conformational changes, but we do see
I see.
the mechanism.
Sure.
And how locally an allosteric mechanism, how we can understand.
I see. So again, I think allosteric mechanisms are a little bit more difficult than the direct effect on the protein. But in general, so this is actually the example I'm providing here is a good example of this. So here, the difference between this leg and this leg is only this phosphate binding here. So you can imagine that could be your mutation, for example. But again, so you need to have the function that you want to describe. You have to describe that function and see what happens to that function if you mutate something. You know, if the protein doesn't change, something else should be affected. Maybe the interaction of the ligand with that mutation site will be affected in your case. Maybe it's an enzyme, the substrate binding is affected. So what is the process that is affected? That's something that doesn't automatically come out of the simulation. So you have to help it with your experimental, with your knowledge about the system. So what is it that is affected by this mutation? Can't say just, oh, this mutation kills the mouse, so what is going to happen to it? No way. You have to have an idea, is it ligand binding? Is it the substrate processing? Is it unbinding of the product? What is the step that is affected? Then you describe it in the wild type case, and then mutate the system, relax the system. If conformational changes happen. If they don't happen, we'll see. And then do the same process for the mutant, the same way that you did it for the wild type. That's the most scientific way of going about it using this technique. And sometimes, of course, it doesn't work, you know, like any other techniques in the yeah. Good. Eight more questions? No? All right, thank you. So we can have the coffee break.